Okay, for the second part of today's lab, we are going to cover microscope use, the anatomy of the microscope, and then we are going to take a look at a whitefish blastula, which is a group of cells that will develop into a whitefish. So we're looking at the very beginning of the forming of an embryo in a whitefish in order to identify phases of mitosis. One of the reasons we use a whitefish blastula is because since it's developing into a fetus, the cells are undergoing mitosis very rapidly. So there's a good chance we'll get to see everything that we need to see. Now, when you are carrying a microscope to and from your desk, it is imperative that you always carry it with two hands, one hand on the arm right here, and one hand on the base. When we work with a microscope, we typically want to start with the lowest magnification and then fine tune our way all the way up to the highest magnification for our class, which is a total magnification of 400. And then that's typically where we will draw and observe our cells and our tissues. So what we're going to do is take a look at this microscope and identify some of its anatomy. First and foremost, as I mentioned, we will always carry the microscope by its arm and the base. Once you place your microscope on the table, we try not to move it because these are compound microscopes, meaning that they rely on two different lenses to magnify the object. And if you drag a microscope unnecessarily, it can cause those lenses to become unaligned, or misaligned, I should say. So typically, we recommend that you move yourself to the microscope or you pick the microscope up and gently place it down instead of dragging it across the table. The next items here highlighted are the objectives and the mechanical stage. The mechanical stage is where you will put your slide in order to view it. Think of it as you are presenting your slide on a stage, a mechanical stage. These hanging down from here, from the head, are called objectives, and they contain the first set of lenses I was referring to in our compound microscope. The red objective, which you cannot see in this picture, contains a lens that magnifies an object by 4 times its normal size. The yellow lens will magnify it by 10 times its normal size, and the blue lens 40 times its normal size. There is a white objective lens that magnifies things 100 times its normal size, but we do not use it in anatomy because we need a special oil immersion in order to use it, and that's something you'll use in microbiology. Now, the other lens is located in the eyepiece. Here we have a trinocular head, meaning it has 3 eyepieces. Typically, though, in a biology lab, you only see a binocular head with 2 eyepieces, and it's within these eyepieces that you have the ocular lens. The ocular lens will magnify the image 10 times its normal size. So to find out your total magnification, you would take 10 for the ocular lens magnification times whatever objective you are using. So if you're looking at the red objective, which is where we typically start, your total magnification would be 10 times 4 for 40. If you're looking at the yellow, it would be 10 times 10 for 100 times total magnification. And if you're looking at the blue, it would be 10 times 40 for a total of 400 times magnification. When you're looking at a specimen, you always start with the red objective, focus it under the red objective, and then you move to the yellow, refocus, and then finally the blue, refocus, and then you would draw. To focus a specimen, you are going to rely on these knobs on either side of your arm. You have a coarse focus knob or coarse adjustment knob and a fine focus or fine adjustment knob. When you are under the red objective, you are allowed to use the coarse focus and the fine focus. We always recommend using the coarse focus first to really get the image sharp, and then you fine tune it with the fine focus. After that, when you move to the yellow and the blue objective, you should only need to touch the fine focus and not touch not touch the coarse adjustment knob. Let's talk about some other items on our microscope here. This nose piece turret, sometimes referred to as just the nose piece, is a dial that you touch when you want to change your objective lenses. We do not touch the objective lens when we want to rotate in between them. We only use the nose piece turret. Here we have the illumination system, or simply known as the lamp. It's going to shine a light through our specimen so we can see the internal workings of our cell or our tissue. Depending on what type of microscope you have an illumination intensity knob. It's located on this model down here. In our classroom, they're located up here. Usually you'll see numbers on it, and we always recommend starting off with an intensity about halfway to the max. Here on the stage, we have what's known as a stage clip. The stage clip would hold your slide into place. And then on the other side of the stage, there's a pole with some knobs that hang down. These are your mechanical stage controls. They allow you to manipulate your slide and move it side to side and up and down, so you can scan your slide for different items on it. There are also some smaller items worth mentioning. The condenser is located under the stage, and its job is to help focus light. The iris diaphragm will help to increase contrast. So if you're having trouble seeing maybe some of the internal structures of a cell, you can adjust the iris diaphragm to really make those shadows stand out and make it easier to see those internal structures. So when you take out or put away your microscope, you should always carry it with the arm and the base. When you are putting your microscope away, you always want to make sure that the red objective lens is down and that the stage is all the way down as well. Again, when you're focusing, you want to make sure you move from the red to the yellow to the blue objective. When you're on the red, you first focus with the coarse focus and then the fine adjustment knob. Once you move to the yellow by touching the nose piece turret, you would only touch the fine adjustment knob, and then again with the nose piece, move to the blue objective lens where you would fine tune with the fine adjustment knob. And then typically we draw what we see in our field of view on the blue objective. When I say field of view, I mean everything I can see in my specimen while looking through a microscope. So we are going to practice today by using mitosis. So before we talk, or I should say before we identify the phases of mitosis, let's quickly review the cell life cycle. So the cell life cycle has two major parts to it. It has interphase and it has mitosis. Now, the majority of the cell's life is spent in interphase carrying out normal processes like producing ATP, protein synthesis, and for our purposes, an important event, which is DNA replication. What does that mean for us? Well, if we're talking about a somatic cell or a body cell, which is another term for somatic cell, our somatic cells contain 46 chromosomes. So if those are going to replicate, we end up with 92 total chromosomes, or another way of saying that is 46 double-stranded chromosomes. This is important because at the end of mitosis, we're producing two identical cells to the one that we started off with. So we have to divide that DNA. And if we divide 46 chromosomes into two new cells, we don't have enough DNA in each of those new cells. So our 46 chromosomes will multiply to give us 92. And then when we divide those into two new cells, our two new cells are identical and each contain 46 chromosomes. So mitosis is really good for growth, like in our whitefish blastula, and repair, so maintaining our cells once we've finished growing. Now, mitosis happens in four major phases: prophase, metaphase, anaphase, and telophase. Let's look at prophase first. During prophase, my DNA is going to start to condense. Up until this point, during interphase, my DNA has been inside of a nucleus. and it's invisible because it's being unwound in order to replicate it or to create proteins. So during interphase, when you're looking under a microscope, you are going to see your cell membrane, a nuclear envelope, and maybe a nucleolus, but you will not see DNA. So that's a good clue as to whether or not you're looking at interphase or prophase. Again, in interphase, you will see a nucleus and no DNA. In prophase, though, that DNA wants to condense, it wants to coil. And the reason for that is it's a lot easier to move DNA when it's compacted and it protects it. If it's uncoiled and we try to move it too much, we could possibly damage our genes. So that DNA is going to start to cluster in the middle of our cell like this. And because we want to move the DNA, the nuclear envelope will disappear so we could move our DNA. So in prophase, you shouldn't see a nucleus and DNA should be visible. Now, our next part of mitosis is metaphase. Oops. I call metaphase the M&M phase because in metaphase, my chromosomes or my condensed DNA will line up in the middle of the cell. And that's to prepare for our next phase, anaphase, which splits our DNA. So during anaphase, the centrioles that we learned about in our organelle mini lecture will start to pull the strands of DNA to either side. So this is my A and A phase. DNA is pulled apart. The last phase of mitosis, which again involves replicating that, taking that replicated DNA and splitting it into two new cells. So taking that replicated DNA and splitting it into two new cells is telophase. During telophase, you'll start to see my cytoplasm pinch off and form a cleavage furrow. My DNA is starting to clump again and uncoil. And because it's uncoiling and these two new cells are getting ready to enter a new interphase, the nuclear envelope is going to reappear to protect that DNA. Now, the last little bit of this is splitting off of the cytoplasm. But since this doesn't involve the actual movement of the DNA, we're going to count it as kind of its own little phase involving the cytoplasm known as cytokinesis. So cytokinesis is our last little step here in splitting our cells, and it's referring to the splitting not of the DNA, but of the cytoplasm. So when you look at our whitefish blastula underneath the microscope, you're looking for these features to identify my phase of my cell life cycle. So again, if I'm looking at interphase, I'm going to see a nucleus with no DNA. If I'm looking at mitosis, I have to figure out if I'm looking at prophase, metaphase, anaphase, or telophase. Or I like to say, don't pee on the mat. In prophase, again, my DNA will be clustered and I should be lacking a nuclear envelope. In metaphase, my DNA will be lined up in the middle. In anaphase, my DNA will be pulled apart. And in telophase, I'll see a cleavage furrow. My DNA is uncoiling as my nuclear envelope reappears, and I may see cytokinesis occurring in the cytoplasm. So let's take a look at what you would see when you look at the whitefish blastula under a microscope. So this right here is a magnification of 400. And you can notice we're seeing a lot of cells here. So we have cells everywhere. Now, cells are 3D, so when we slice through them, sometimes we don't catch exactly what we want to see. For example, this cell here looks empty. I don't even see a nucleus, and that may be due to the fact that we didn't section it right where we wanted to. But other cells you can see. For example, here, this cell has a nice, clean nuclear envelope and a little nucleolus in it. That's it. So that tells me that's interphase. If I look here, I have a cell membrane and I have a dark clumping of DNA. That tells me I'm looking at prophase. Here I have another cell where my DNA is lined up in the middle. That would be metaphase. Here I have a cell where my DNA is being pulled apart. That would be anaphase. And then here I have a little cleavage furrow between two cells where my DNA is starting to clump in either cell. And that's telling me I'm looking at telophase. And if you look closely, you can even see a little bit of cytokinesis occurring, that building of a cell membrane in between those two cells. Okay, I hope this helped you to become a little bit more familiar with the microscope as well as mitosis. If you have any questions, please feel free to contact me.